Last time I mentioned p-value, e-value, z-score, what they mean. To repeat those again, p-value is the probability. Okay, so therefore it's between 0 and 1. It's the probability that you get the same or better alignment, meaning that you achieve the same or better score by chance. Can you get a similar score, a same or better score with a random unrelated unrelated sequence? That's the p-value. And e-value is the number of such random sequences in the database that could have achieved same or better score. So you understand the difference? The first one is the probability. The other is the e-value is the number of it's the number of random sequences that could have achieved. So also when we are computing the e-value, what we need to take into account the database size as well. This number, having random sequences, having the same score by chance, that number can increase if you have a larger database. Okay, if you are, the first one is just talking about probability. There is no number associated with it. The probability is this. But imagine you apply this probability. For example, if the probability of being colorblind is 10%, in a room with 20 people, that 10% evaluates to 2%. Okay, if you convert it to like an e-value type of that difference, 0.2 probability is converted to number 2 in a population of 20 people. But in a population of 2,000 people, that is going to evaluate to 200. Okay, you may get two different e-values, 2 and 200, when the population size changes. E-value is something similar. Based on the database size, the e-value can change. And z-score is the number of standard deviations you are above or below the mean of the population. So how much this score is different than the average score. And when we talk about average score here, average score between unrelated sequences. If I just randomly align all the sequences in the database and I get alignment scores, what this gives me is that a measure of what is an alignment score on the average between completely unrelated sequences. What's an alignment score look like? Is it 0 all the time? Do you get 0 alignment score between unrelated sequences? Probably not. Probably if you align completely unrelated sequences, you can still get a positive score. But what does this positive score look like? Is it 10? What is it on the average? 10, 20? So if you get a feel of how the average alignment score looks like between completely unrelated sequences, then if you have an alignment which has a higher score, saying that, okay, I have an alignment which gets a score of 100, then the z-score is how much this 100, how much standard deviations away from the average of these unrelated alignment scores is this. So a z-score of 7, 10, 12, 15 may mean a good thing, but again, there is no standard for z-scores as well. Z-scores sometimes for some problems, a z-score 7 may be considered a good thing, but there's also statistical theory behind it, which z-scores are good, but it may, for certain problem domains, it may have a different meaning. Okay, so this is what I already told you about p-value, e-value, and z-scores. So e-value can also be considered the expected number of sequences that will produce the same or better score by chance. And do not forget that the expected number of unrelated random sequences. Okay, because we do not know in the database which are really related. In the database, out of all the sequences in the database, what is the expected number of sequences that can produce same or better score by chance? And BLAST algorithm produces this e-value. And these both p-value and e-value, the smaller they are, the better, the more significant the alignment is. However, for z-score, the higher the z-score, the more significant the alignment. Okay. Now, how do we compute this e-value for a given alignment with gaps. Okay, if somebody gives you an alignment with matches, mismatches, and gaps, how do you compute an e-value? The first thing is we need to. This was real difficult. I mean, they need to work on this computation a lot, and finding a closed form formula for e-value for gapped alignments was very difficult. It was difficult at first. So they first started with the simpler problem. Instead of working on a complete alignment with gaps, matches, mismatches, and computing it. What is the probability of having such an alignment? Do we have it by chance? So the goal is to answer that question. Okay, if I give you an alignment, and your goal is this alignment? What is the probability of having that alignment between two unrelated sequences? So, is it a good thing or a bad thing? So, in terms of can we achieve it by chance or not? So they first tried to analyze this and found out that finding a probability as a formula, what is the probability of having this alignment as a formula between gapped in an alignment which contains gaps is an extremely difficult task. Okay, mathematically it was very difficult to find out a formula for this. So they started with a simpler problem. First, Erdos and Reni, the first problem they attacked was, what is the significance of a match run? What is a match run? Match run is this. In an alignment, you have matches, right? A match run is a consecutive series, is a series of matches, one after another, no mismatch, okay? If in an alignment, for example, if you see 10 matches of amino acids, or 9, 7, or 11, 12. So a consecutive, a series of matches in an alignment is named as a match run. Okay? Now, actually, it turns out that we can compute the exact probability of observing what is the probability to have 10 amino acids match. For example, if I have these or let's think about DNA sequences. Okay, well, I'm going to use a better pen. Okay, now imagine I have 1, 2, 3, 4, 5, 6, 7, 8. Okay, an 8 nucleotide DNA sequence, and I'm going to I have a sequence generator, okay, which spits out some nucleotide sequences one by one. Okay, it's just constantly spitting out these A, C, G, T. Okay, we are observing there's a constant stream of DNA sequences coming out, and in this constant stream of DNA sequences, what is the probability that a sequence of length 8 is going to be this A, G, C, T, A, C, C, A. Okay? What is this probability? This is, we are going to first start with this. So this is the significance of a match run. What we have here is a matching run, a matching of 8 nucleotides. We have a match run here. And now let's try to go over the slides. We are going to compute this probability exactly. We'll be able to do that exactly, and we are going to talk about what is the and we are going to extend this probability to, for example, this small sequence may be part of a very long sequence. So in this very long sequence, what is the, maybe we may not get an exact match for this exact sequence, but we may get a matching run of length 8 in somewhere else of this sequence. So we are going to analyze that. And then the significance of this matching run was extended by Carlin and Altschul, who are also the developers of the BLAST algorithm. They computed the significance of local alignments without gaps. So they extended this matching run to include mismatches as well. So if we can also compute, if we have a scoring matrix, we can think about these matches as a certain score, and we can allow for mismatches within this matching run, and we can compute the probability of a local alignment without gaps. Okay, local alignment without gaps means that it has matches, mismatches, and the probability of that local alignment without gaps can be computed exactly, which uses the theory behind scoring matrices a lot. So this analysis, this kind of an extension, made use of the scoring matrices. Instead of counting just matches and mismatches, ones and zeros, it made use of the scoring matrices. So therefore, given the score of a local alignment, this matching run has the measure we have about the matching run is its length. Okay, a match run of length 8, a match run of length 10. Okay, when we're computing this probability, the probability is going to decrease as the match run length increases. It's more difficult to get a matching run of length 20 compared to a match run of length 2. Okay, length 2 you can get by chance. Maybe that probability is not that very low, but getting a match run of length 20 would be very difficult. The probability is low. And here, when I'm talking about this probability, I'm talking about lengths, as you can see. I mean, length of 2, length of 20. Now, when we talk about scoring matrices, we will extend this definition of length to a score. Okay, what is the probability of obtaining a match, an alignment, a local alignment, aligned region with a score 20, with a score 25? So we are going to be extending the length to the score measure, and then this definition was later extended in BLAST 2. That was the main reason in BLAST 1. One of the main reasons, which did not, the original BLAST did not have the gaps. It was doing ungapped alignment, and one of the main reasons, I believe, I'm not sure for it, but I believe it's this. I mean, because the statistics was not studied. Oh, sorry, but this is my cell phone. I forgot to put it to silence. Okay, so in BLAST 2, they were able to do gapped alignments because they studied they extend the significance measure to include local alignments with gaps, okay, which was much more complicated. And I'm not going to go into details, all the mathematical details of this one. We are going to probably stop here in the context of this course. Significance of global alignments also. Computing the e-value by using a certain equation, closed form formula, will be difficult. And what FASTA does is, it does not use a closed form formula like BLAST. FASTA uses empirical evaluation of these significance values by having a distribution of scores of random alignments. By looking at these score distribution of random alignments, how they look like, they compare the computed alignment score against these empirically previously computed score distributions. This is not what BLAST does. Now, let's start with the significance of a match run with a very simple example. Okay, forget about DNA sequences. Let's start with, again, it's like sequence, but with just 2 letters. Heads or tails, H or T. Okay. Now we analyze. We are tossing coins, and the coin can come either heads or tails. And imagine we perform an experiment which consists of this many coin tosses, and the black circles here indicate the coin tosses which came heads. Okay. Now here. For a fair coin, the probability of observing a head in a single coin toss is 0.5, and similarly, the observing of the probability of observing a tail is also 0.5. Okay, we either observe head or tail, and these coin tosses, they are independent from each other. All these experiments are identical and independent from each other. A coin toss, your 5th coin toss is independent of your 4th coin toss. Whatever it came at the 4th coin toss does not influence what you have, your experimental result at the 5th coin toss. They are identical and independent events. Now, therefore, to get 5 heads in a row, I mean, you are going to toss the coin 5 times, okay? And you are going to get heads in all these 5 coin tosses. That probability is 1 over 2 to the power 5. Okay, that's a low probability. So the first one is going to come heads, the second also heads, the third also heads, fourth heads, fifth heads. So 1 over 2 times 1 over 2 times 1 over 2. This is 1 over 2 to the power 5. It's approximately 3 in 100. Okay, it's that 3% chance you can get 5 heads in a row. Now, however, imagine if we ask the question in a different way. I mean, in other words, it's 3% chance I am going to get 5 heads in a row. So it means that if you perform this experiment a lot, okay, if you toss the coin 100 times, during these 100 times coin tosses, you're likely to expect 5 heads in a row, okay? Because I mean, if you do this experiment once, if you just throw 5 coins, the probability is low. However, you are doing this 5 coin tosses. For example, imagine you started from here, okay? You get a head, okay, right, great. At the second coin toss, oh, you don't get a head. Okay, let's start the experiment again from scratch. You start from here, you get a head, another head, oops, no, tails. Let's start again, oops, tails, no, let's start. So we can repeat this experiment of observing 5 heads anywhere we want. Whenever we fail, we can start it again, okay? If we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 14. So here, it's written here. So in 14 coin tosses, there are how many different places you can start this experiment? You can start at the first coin toss and the 9th coin toss, but you cannot start a new experiment at the 10th coin toss because there's only 4 left. If at the beginning somebody tells you you have 14 coin tosses, so in the number of experiments, 5 head observation, that experiment you can try is actually 14 minus 5 plus 1, I guess. So it's 10. We can start this experiment from 10 different places. Let's see. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this is the last time I can start this experiment. Okay, so it's similarly, if you're going to do m coin tosses and you want to observe L heads, so it's like m minus L plus 1. You can try this many times, this experiment in m coin tosses. And the expected number of times that 5 heads is going to occur in this above 14 coin tosses, because we performed the experiment 10 times, and the probability of observation in a single experiment, our probability was 0.03, but we performed this, we tried this 5-head experiment 10 times during these 14 tosses. So the expected number of the occurrence of 5 heads is 0.31. Okay, this is what the e-value of this experiment is. So in other words, if you perform this 100 times, you would probably see, expect to see 3 5 heads in a row in these 100 coin tosses. So the e-value is not a probability. Okay, e-value here, this 0.3 is not a probability. It's not like your 0.3 probability you are going to observe 5 heads in a row. It's not a probability, okay? You should not treat the semantics of this e-value that it's not a probability, it's a number. Although it is a floating point number, it's a number less than 1 here, 0.3 is something less than 1, but it's not a probability. And if this number of coin tosses was 100, if we did this 105 times or 104 times, if we did it 104 times, the expected number would be 3.1. So in those 100 coin tosses, you expect to see 3 times 5 heads in a row, okay? So it's not a big deal if you throw it 100 times. It's not that small probability, this becomes something observable. And if we convert this to formulas, okay, the expected number, so this expected number, this 3.1, can be formulated as the length L run of heads. So if the probability of head is 1 over 2, the probability of observing a length L run is going to be 1 over 2 to the power L, if we are looking for a L heads, L consecutive heads. And this was our probability, so it's P to the L. And the number of times you perform the experiment is exactly n minus L plus 1, okay? The exact E-value is this. Our E-value is n minus L plus 1 if we are going to throw the coin n times. This is the number of times we are going to perform this matching run L experiment. This is the exact number, but in general, this L is very small compared to n. Okay, for example, when we are looking for local alignments, the length of the local alignment is usually very small compared to the whole length of the sequence. Therefore, this can be approximated. This E-value, we can ignore this part and just write np to the L. Okay, this is an approximation which ignores that when L is small compared to n. And we can work out the equation in the reverse way, the inverse problem, which states that what is the minimum? We can ask the different question. How many times you need to toss the coin so that observing 5 heads in a row is not something you are going to expect? Okay, so how many times you need to throw the coin so that you expect to observe 5 heads in a row? So now what therefore we write this equation. So if the E-value is 1, okay, the expected, if you're interested in observing a single, the probability of observing single 5 heads in a row in n, so we want to, so this is, actually this is something different. This question even is not the one so in order to answer that question, this P to the L, we need to take n out, okay? So the number of, so in that case, this is trivial, okay? N is going to be 1 over P to the L, okay? If you want to observe 5 heads in a row once, you need to throw this many coins so that 5 heads in a row, observing 5 L heads in a row is something expected. But usually we do not have a chance in biological sequences. We do not have a chance to increase or decrease the length of the sequences. The sequences, biological sequences, are found and they do not change. Okay, what I'm saying is that the sequences are given as input. In this alignment, we have 2 protein or DNA sequences which are aligned, so their lengths are known. Okay? The question that we ask in sequence alignment is that if you have sequences of length this and length that, and you have seen in this alignment a matching region between these 2 protein sequences, and you look at this length of this matching region, is it something that you expect that can happen by chance or not? So we ask this question, therefore, what is the expected length R? Okay, of the longest match in n tosses. So if you throw the coin 50 times, for example, what is the longest match run of heads that you would expect to see once? So that it's not surprising. So, and therefore, in order to find that, we use, we can take the log of both sides, and then this length R becomes log 1 over P base n. Okay. So this is the, I guess I have an example here. So here, this formula was developed by Erdos and Rainey, and it's, if there are nth rows, then the expected length R of the longest run of heads is given by this formula, and it's derived directly from this. And here is an example. So if we throw the coin 20 times, okay, and somebody tells you that, okay, I throw the coin 20 times, and 4 heads in a row, I observe 4 heads in a row. 4 heads in a row is very low probability, okay? 4 heads in a row, so I'm very lucky if somebody tells you that. You should, if you know this statistics well, you should say, okay, you're not lucky at all. This was expected, okay? This is what I would expect. If I do it myself 20 times, I would also expect to see 4 heads, okay? 4 heads in a row in some place, okay? So this is what this formula is used for. Here, log 1 over 0.5. So this P is probability of observing heads. So 1 over P is 1 over 1 over 2. So in log 2 base, log 220 becomes 4.3. So the expected number of heads that you, so we expect a run of heads of length 4.3 once. So this is how you read this. Okay, the expected number of observing 4.3 heads in a row is 1. So it's, these floating point numbers used for counts may be confusing, but you should get used to that if you are going to use e-values. Now, okay, this is, how can we extend this head thing, which is just a single sequence of heads or tails, to an alignment, okay? How do we model DNA or amino acid sequence alignments as coin tosses? And it's not going to be very difficult. We are going to make use of the alignment matrix. The observing a head, we are going to replace it with observing a match between 2 amino acids, okay? This is what we are going to do, okay? You toss the coin, you get a head. Similarly, what's the probability that you pick this amino acid and you pick another amino acid? What's the probability that these two match, okay? Let's think about that, okay? If I tell you that this is my sequence and this is the amino acid I have, if I pick randomly another amino acid, these are amino acids, okay, 20 letters, an amino acid has 20 letters. If I have another sequence and I pick a random amino acid from the other sequence, what is the probability that these two match? Come on. 1 over 20. Yeah, that's correct. That's the correct answer. The wrong answer would be 1 over 20 times 20. Okay, meaning that, okay, 20 letters, this one, the probability of observing A is 1 over 20. And the probability of observing A here is also 1 over 20. So the probability of this match would be 1 over 400. That would be the wrong answer because the thing is that we did not ask what is the probability of having both of them as A's. If I ask the question, what is the probability that if I pick 2 random amino acids and it turns out that they are both A's? If I ask that, that will be 1 over 400. But one of them is already given as A, okay? Given that one of them is an A, what is the probability that the other is A is going to be, I mean, this other one can be one of these 20 different amino acids. So the probability of match is 1 over 20. In other words, these match could have been, I mean, the probability of observing 2 A's may be 1 over 400, but a match is not only considered between 2 A's, it may be 2 C's, 2 D's, 2 E's, 2 F's. So there are 20 different such matches. If you multiply 1 over 400 by 20, you get the probability of a match is to be 1 over 20. And for DNA, it's going to be 1 over 4. So and how many times we are going to perform this experiment? If we consider the coin toss idea, how many times we can get, if we think about matches between two protein sequences, I can get a match between here and here, or I can get a match between here and here, or here and here. So I can get a match between all pairs of amino acids. So therefore, we go to this, the number of experiments we perform is here on this table. If we are interested in a single match, what is the probability of obtaining a single match between these amino acids? These are the experiments, these are the places that we can observe these single matches. We can observe a match between the last amino acid of this one, between the first amino acid. So these dots, they show the matches, and these squares, the size of this matrix shows me the places where I can observe a single match. Okay? A match can be observed between all pairs of amino acids between them. Now, however, if I ask you, what is the probability of observing 3 matches, consecutive 3 matches? Then we cannot just combine matches that appear different places. Three matches, what are they going to be? They are going to be diagonals, right? These 3 matches on the sequence, what is the probability of observing a match of length 3 between these sequences? Here, in that case, that match can be this, but it cannot start in the last 2 amino acids, because in the last 2 amino acids the length is not enough. If the third amino acid from the end is not a matching, then you have no chance to get the length of a match of length 3 in the last one. So you are going to look at all the possible diagonals it can start. So all the diagonals, again, at these endpoints, we are not going to be able to have the diagonals. So any diagonal that appears anywhere of length 3 is a possible way we can try this experiment. So again, ignoring the length of, even with this length 3, compared to the length of these amino acids, this can be, the fact that we cannot start a match at these endpoints can be ignored. Okay? In other words, if I have a length m and length n sequence, if this m and n are large, like 100, obtaining a match of length 5 is still going to be, if this is, for example, if this is 100 and if this is 200, for example, I can say that I'm going to try this experiment in 200 times 100 places. I can start this experiment. I need to subtract this 5, and not only 5. I cannot figure out the exact formula now, but this number that I'm going to subtract is going to be very small compared to the whole matrix places. And again, we can ignore and approximate the possible number of, we try this experiment by the length of, by the multiplication of the length of the sequences. Okay, so the probability of an individual match is 1 over 20. So the expected length R is going to be log 1 over log 20. And the number of times we tried the experiment is going to be, so this n is going to be m times n. Okay, this is the number of times we tried the matching experiment. And this is the expected local alignment with perfect matches. The length of the maximum length of the local alignment with perfect matches that you would expect to see once in 2 sequences of length m and n. And this is described in detail in these following slides. If we are considering only single matches, we expect to see 4 matches in these 2 sequences. An expected number of 2 successive matches can be approximated. We can ignore the fact that we cannot perform the experiment at the boundaries. So it's 10 times, 8 times, again 0.05, 0.05. So observing 2 successive matches as the number is smaller. Okay. So I'm going to stop here for